Hi, all. Welcome. Thank you for being here. I'm going to go ahead and mute everyone. So kindly stay muted while the presentation is going on. We'll give a few minutes as people come in. Hi, welcome everybody. We'll get started in about a minute. Okay, I'll go ahead and get started. So thank you all for being here for our part 2 of how to secure research funding. Today, Dr. Wong is going to talk about writing your grant application, and I'll start with just a couple of small housekeeping things. So like I mentioned, please stay muted during the presentation, and if you don't mind, please use the Q&A box for questions. So we'll be looking there for your questions and take questions at the end. I want to really thank Audrey Huang for being back to talk to us again about research funding and today about writing your grant application. Audrey is the Director of Science Communications for the University of Maryland Center for Advanced Research and Training and Innovation. And we so much appreciate her sharing her expertise with us. So Audrey, I'll hand it over to you.
Great. Thank you so much for having me again. And it's always a pleasure to share ideas with folks who are hungry to launch their research careers. So thanks for showing up today and spending the time. I wanted to figure out my mouse here. Here we go. So I know we designed these as a series, but also they're meant to be standalone. So if you didn't show up for the last one, don't worry, there's not going to be a quiz for this one. However, I did want to spend a few minutes just going through a little recap of what we talked about a few weeks ago for those who missed it and for those who were here and just need a refresher. We covered how to find sources of funding for your work. We talked about funding cycles in general. We talked about NIH specific funding mechanisms. I gave you a couple examples about how early faculty members have diversified their funding portfolios and kind of staggered timing and source of funding to launch a steadier stream. And then I ended pretty quickly with aspects of good grantsmanship and the importance of focusing on your audience, which would mean reviewers and not anybody else. So I've had these highlighted because I actually wanted to go through them again because I think they're critical as an introduction for this week's topics. So, grantsmanship. I know we're talking about how to write a grant application, but it's really the entire aspect of grantsmanship that I want you all to think about and continue thinking about as you launch your careers. And it's not just the writing. The writing is critical. We'll cover that, but it's really the big picture. So, absolutely, you need a clear and focused scientific idea. You have to present your proposed research in alignment with the funder's priorities. And that means it will change. It should change your research proposal, whether you're applying to NIH or a private foundation, or even to an internal institutional grant at your home institution. So this is a really critical aspect that I think a lot of people don't spend enough time thinking about when they repurpose drafts and grants. Your proposal should be well structured and readable. There should be absolutely a very strong specific aims page. We will talk about that in more detail today. Your proposed research should be rigorous, but feasible. It should not be so rigorous that it's like impossible. You should be able to do it, and you should be able to do it in the time frame of the funding you're applying for. So if you're applying for a 1 year little quickie grant, you should have results by the end of that year, as opposed to if you're applying for a 3 to 5 year R01 grant, then the proposed work should take up that amount of time. You should have a strong team and research environment. And so that goes beyond you. If you're applying for career development awards, that means your mentorship team. If you're just starting a lab, it means your collaborators and your staff in your lab. The environment, your home institution needs to have the resources and needs to support you. Talk a little bit about that today. You have to have the ability to anticipate, review, or critique. And this comes with getting your drafts done in time to get good, solid, and deep reviews from colleagues and friends. The more you have other people read your work before you turn it in, the better that work will be. You have to have a compelling narrative. So you need to be able to tell a story, and your writing needs to be polished. I highly recommend, I said this last time, if you are not a native English speaker, to find a resource, whether it's a paid editor or a very good friend who is a native English speaker and strong in grammar and writing, to help you with the polishing of the final draft. And then last but certainly not least, administrative accuracy. And this just means, are your margins, your fonts, your budgets, do they add up? Do they make sense? All of these little details matter. People look at them very carefully, and I have seen, unfortunately, grants get turned back immediately for administrative reasons, and that is putting the wrong information in the wrong section, or having your margins too narrow. It gets turned around immediately. Nobody looks at it. You're out for that cycle. So don't have that happen to you. All of this is very critical. So it's not just about the writing. The writing is very important. We'll cover that today, but all of this matters. And then the other thing we covered last time is really know your audience. So I'm going to be talking a lot about NIH grant structure today, because I think that is it's still the bread and butter of historically of how basic biomedical research is funded in this country. And so until we have reason to believe that's going to change, I think it still will be the bread and butter for you all as you launch your careers. We're just going through a phase right now where things are changing. But know your scientific review officers and study section members. They're all online. Understand who the experts are that you're writing for. You're only writing for the reviewers. And that means you're not writing for your mentor. You're not writing for your department chair. You're not writing for your current lab colleagues. You will ask them to review things, and they will have opinions about how you, especially your mentor, how you're designing your work. You have to be able to separate that out from what the reviewers will say. Now, granted, your mentor has more experience than you. They will have had more experience in their professional career with reviewers and study sections. And so for that, they are valuable reviewers, but they are also conflicted because you're their trainee right now. You're taking some aspect of the work you're doing with them. You're going to launch your own career. There may be competition, there may not be, depending on how you've designed this agreement with your current mentors. But know that they're conflicted, and you need to always keep your eye on the prize and write for the grant reviewers. Okay, so when it comes down to it, however, the reviewers will only fund proposals that they understand. So straight right across the board, there's going to be immediate rejections for administrative reasons, and then there's going to be rejections for grants that are not aligned with the study section or the funding organization they've been submitted to. So don't be in that pile. And then as the reviewers consider more carefully, if you've made those first couple cuts, as they consider more carefully your grant application, they need to be able to understand it. And that's what we're going to cover today. So I'm going to backtrack a little bit and talk a little bit about making writing a habit. I think this is absolutely critical. I think for those of you who are clinical and in the clinic sometimes and doing research, and your time is split already versus those who are basic science and are at the lab or at a computer. Um, it's hard to shift gears in your head and energetically and sit down at a computer and write. There's always something you could be doing. There's always something else, another patient you could be seeing. There's always another paper you could be reading, somewhere you could be going to. Making writing a habit is very important. We'll get to that. And then we're going to cover how to define your research project and write your specific aims page. We're going to talk about significance and innovations, which are a critical aspect of the research plan. We're going to then talk about the rest of the research plan, where you're proposing the experiments that you're going to do. We're going to spend a little bit of time talking about your biosketch, because in my experience, I find most people spend the least amount of time on their biosketch and it dings them. So we'll talk about that, and we'll talk about use of figures and illustrations in your application and what to do, what not to do. Okay. Making writing a habit. Like I said, I know it's hard. For those of you who don't naturally write, for those of you who have a bazillion things on your plate, for those of you who have split time between clinical and research, it is almost impossible to sit down and find quiet time to write, and never mind developing a habit. I'm going to say, do it now, do it even if you don't have a grant in mind, make it a habit to write. Schedule time to write, have a start and a stop time. You don't set out to run a marathon and not train for it, right? You do a couch to 5K, you do a couch to 10K, you maybe try a half marathon, and then months down the line, you pick a marathon and you work your way up to it. Same thing. You cannot write an NIH grant from zero if you've never sat down to write before. You can't write a full manuscript from zero. You need to get in the habit of writing. You also should stop yourself as you're practicing and setting a habit, because the worst possible thing you could do to yourself is make yourself sit in front of a blank screen for 4 hours at a time. You are wasting your time. You're right in thinking that, and you need to learn to be more efficient. So set a timer to start and set a timer to stop. This is why, for those of you who use the Peloton app, Peloton is brilliant, because you're not signing up necessarily for a 1 hour exercise class. They have 5 minute classes, they have 10 minute classes. Guess what? You can do ab work in 10 minutes, right? You can squeeze at any time. Same thing goes for writing. If you're deliberate about it, you can sit down and write quite a bit of stuff in 10 minutes. The thing is, you need to remove all your distractions. Don't check your email. Turn off all the dings and whistles and buzzes and all that stuff. Take off your smartwatch and remove all your distractions and just focus on writing. And then when your timer for your writing goes off, then you stop and go back, do what you were doing before, and do it again tomorrow. Don't self-edit. What am I doing? I just lost my screen. Don't self-edit yet. So keep everything that you're writing as you set up a habit. And I would start with the easy stuff. Write what you did that day in the lab. Write something. Write your materials and methods. Write ideas about, your ideal research. Just write, and then stop after 10 minutes. Stop after 5 minutes. Stop after 10 minutes. Bring it up to 15. These are some books that I recommend that may or may not help you if you're into reading about writing. The most useful one actually is the WAG Your Work. If you don't have a WAG, and I don't know if you do at NIH, somebody should start one, or you should start several. If you need more structure to help yourself write, join a WAG, a writing accountability group, or form a WAG. And the WAG Your Work website or book will tell you exactly what you need to do. You need at least 4 people. You need to commit to an hour a week for 10 weeks. And by the end of 10 weeks, there's a recommended structure agenda that you can follow. Anybody can follow. It doesn't have to be, you don't have to be the same subject matter. You don't even have to be the same areas of research. It's just at least 4 people who are committed to starting a habit and a writing accountability group. And this is just data that the woman who published WAG Your Work, Kim Skorupski, has published, which she has noticed, before WAGs, most academics are spending more than 2 hours, or at least more than 1 hour. That would be most, the purple, sitting and writing and laboring over their writing. After WAGs, after they've established a habit and learned to become more efficient, more people are writing in less time and getting just as much done. So totally useful. We run them here at University of Maryland School of Medicine. A lot of academic organizations have formal and informal WAGs. Just gather your friends if you don't have formal ones and get something started, because it provides accountability and just some structure that you might need. Okay, enough about writing. Write often, write. Okay, let's switch topics. So defining your project hypothesis and writing your specific aims. So last time we talked about the life cycle of grants. This time I'm going to talk about the life cycle of a research project, but this is all very familiar to you all. You have an area of interest, you review the literature, there's an exclamation point under there because I'm going to come back and harp on this for the rest of the next hour and 10 minutes that we have. Reviewing the literature is so critical. You're going to have to do it over and over again. It's a huge deficiency I see in a lot of early stage grants. And again, it's going to come back up. You're going to identify a gap in the field that you're interested in pursuing, answering, solving. You'll develop a research question. And then from there, you'll generate a focused hypothesis once you dig in a little bit more. And then you'll define the experiments, or somebody has defined the experiments for you as you've progressed along your training path. You'll conduct the experiments, analyze data, draw conclusions, advance science. And as you're drawing conclusions, you'll find more gaps and you'll go back and review the literature and pursue those gaps. So it is a cycle. And each time you need to figure out, each time you have that interesting question, you're going to have to go figure out, well, what else has anybody else done about this or not in the past? And how have they solved it? How have they adjusted? Have they addressed it well? And you will only know that by reviewing the literature. Okay. So review the literature for your own edification, but also consider writing a review article in this field if you're going to delve into it with any depth. So if you're going to read a billion papers about your area of interest, do it, but do it in an organized fashion and get a publication out while you're doing it. And let's see. Oops. My point here is not for you to stare at a lovely beach and that deserted tropical island, but my point is that you are not, none of us are operating in isolation on a tropical island. All of science, all the research that you're doing is related to something that somebody else has done, somebody else is currently doing. You're not in a vacuum. And so the most common, like, first grant pitfalls I see are drafts that are not aligned at all with funders' goals. And how do you fix that? You go to the funder and you see what they have supported previously and make sure that your question, your research project, your research question fits into their overall scope of the mission of the funder. Now, National Institutes of Health writ large, I see a lot of people drafting grants that are so basic science, they never come back to human health. which is the mission of the National Institutes of Health. It sounds very basic when I say it out loud, but you can't imagine how many first drafts I see that are so focused on the science, they forget about the patient, the wellness, the health, the human. Also, first time grant writers, early draft, first grants, poorly defined innovation and impact. How do you fix that? Read what has been published previously and work that into your draft. How is what you're doing new and innovative? Well, you can only compare that if you know what's been done before, right? How does it impact human health? Well, you can only really know that if you've looked at what's been done before and you have the adequate citations. And then the last thing is failure to demonstrate significance. We'll talk about significance, innovation, and impact next, but again, read what has been done previously. Why is what you're doing now, what you're proposing to do now, so significant? What are the gaps that prior research, not necessarily done by you, but in the field, what have they left unanswered and open for solving? And you need to absolutely know that in order to be successful at this. Okay, so again, not on an isolated tropical island. You are your work is a tiny little cog in a massive, massive machine of history, of scientific research that's currently ongoing. What you need to be able to do in a grant proposal is to describe your project not in isolation, but describe your project in the context of all these other cogs in the machine. Now, you don't have to be exhaustive about all the other cogs. Part of good grantsmanship is knowing which cogs you need to refer to, no matter whether they're proximal or distant, to make your point that your research will be significant and innovative. Right. And that comes with understanding what has been done in the past in the field and what's going on maybe in other fields, related fields, and being able to parse out how your work fits into that greater picture. So defining a research question, I don't know who came up. Well, I do know this acronym comes from this book. I have not read this book in full disclosure. However, if you remember FINER maps, which is not something that rolls off the top of my tongue, you'll remember that those are all aspects of a good research question. So it needs to be feasible, interesting, new, ethical, relevant, manageable, appropriate, has potential value, or it's publishable, and systematic. This Designing Clinical Research book is not just for clinicians. It is now in its fifth or sixth edition. I think it was first published in I want to say the 1970s. It has been around for a long, long time. This fifth edition just came out in 2022. It may be useful for some of you all who are inclined to read books for tips. Okay, so just getting back to the life cycle of the research project, we've reviewed the literature, or you think you've reviewed the literature, you think you've identified a gap in the field, you now want to develop a research question and generate a focused hypothesis, okay? So defining a research question, this has to be something that can be answered. And should be a new question, okay? It could be a specific aspect of a broader topic that's been worked on quite a bit, but it should be a new aspect that has not yet been looked into, or not yet been looked into in the way you want to look into it. And again, not to beat a dead horse, but review the literature, make sure that you know what's been done previously, that the new question is new, that it maybe somebody's, and this is a tricky part, if it's not reproducible, right? If it's turned out to be a negative result in the past, it will not have been published likely. So how do you figure that out? You also, I talked last time about networking at conferences and, identifying mentors in your field, which is useful both for your own research progress as well as your career progress. But you have to talk to people and pressure test your question. You have to understand, what are the standards of methodology, study design? How have people answered similar questions in the past? How have people studied similar problems in the past? What have they measured? How have they made advances? What worked? What didn't work? And then what is there still left to answer? And you need to be able to basically pick that out and make it clear. Okay, part of developing that research question is assessing the rigor of previous work, previously published work from other people, previous work in your own lab and environs. And this will help you define those knowledge gaps to identify exactly which avenues you want to pursue and streamline and fine tune your research question. So, size of previous studies, were they small? Were they pilot studies? Smaller studies, typically pilot studies, are less rigorous in the sense that somebody just wanted to see if they could see an effect at all. Now, if they saw a positive effect or if it was promising, yes, the next step would be to take that and scale up. Scale up, make it more complex, ask the same question in a different way, drive try to drive deeper meaning from the answer. Um, what is it about previous studies that perhaps weren't sufficient in answering the question? Randomization, was there appropriate randomization in clinical studies, for example? Blinding, was there adequate blinding for the assessment to be unbiased? This is talking about clinical work. Sample size, statistical methods, were the statistical methods appropriate for the sample size? Was it powered appropriately? Incorporation of relevant biological variables. Were the previous experiments well controlled? Were there uncontrolled variables? Are there more variables we could look at? Are the key resources authenticated? Were the appropriate number of relationships tested? Are the specific outcomes the right ones to measure for answering this question? Was the right patient population used or the right animal model used? Could there be a better patient population or a better animal model? Have things improved since the last conclusions were made about this field? And then lastly, how were data handled? How were they collected? Were the inclusion, exclusion criteria sufficient? Did they make sense? Was there bias? These are all opportunities as you're reading the literature to identify things to ways to push the research further in your field of interest. So some just examples of research questions. And the reason why I do this is because you need to be able to do a big picture research question as well as verbalize what your specific, really fine-tuned research question is, because you're going to be talking to different audiences in your research career about your work. And so sometimes it's not appropriate to talk about specific genes and names, upregulation, downregulation, that sort of thing. And sometimes it's more appropriate to ask questions like this. So go through the exercise, even if you have a very narrow and fine-tuned question, back it out, and can you make a very lay version of your research question accessible? So these are just some examples. Why do some endurance athletes die of aortic dissection? How do bacteria become drug resistant? Which antibiotic or anti-diabetic drugs work best for which patients? And what you need to do when you're starting to think about these research questions is then, of course, I'm going to harp on this, go back in the literature. What are the actual gaps when it comes to aortic dissection in really fit individuals? What do we not know yet, right? What do we not know about GLP-1 receptor agonists? They're sweeping the nation, but do all of them work for all people, or do some of them work better? Like, what aspects of the insulin pathway are being impacted by these receptor agonists or other types of molecules? And so that's where you need to start asking deeper and deeper questions and start refining the research question, especially for your grant. So in the field of athletes and aortic dissection, well, it turns out TGF beta signaling is very important for vessel health and vessel integrity. But it turns out that there are conflicting data out there as far as TGF beta signaling. So how is it that TGF beta signaling is both good for and bad for aortic health? How does it protect against and promote aortopathy? Um, we don't know this. How do bacteria become drug resistant? Well, drug resistant, let's look at more specifically. We know some of the genes that are involved in mediating drug resistance. We know there's a ton of bacteria out there. So this is when we start looking at specific strains of bacteria. Staph aureus was recent, it's just recently been identified. We don't know very much about it. It is sweeping the nation in a negative way. We do know that it expresses genes for beta lactamase and also penicillin binding proteins. We do know that it becomes desensitized to lactams and lactam class antibiotics. So maybe these genes are being the transcription of these genes is being altered somehow to mediate Staph aureus resistance to lactam, so beta lactam drugs. So maybe the question is, how are they being, what's changing the regulation of these genes to mediate the drug resistance? And then anti-diabetic drugs, well, are there genetic markers? Can we start looking at specific players in the GLP, GIP, insulin, and all the regulatory pathways in metabolism to start looking at can we predict which drug works best on a patient before they spend the money, $1,000 a month or something, to pay for these drugs? So, because you didn't have enough acronyms with Finder Maps, there's another acronym called PICO, developed by another group, which helps you identify variables to measure, particularly for clinical research questions. And PICO stands for population, who are you studying, exactly which population, intervention, what is the treatment, and what are you testing? Comparison group, are you testing diabetics to non-diabetics? Are you testing well-controlled diabetics to uncontrolled diabetics? So who are the populations and how are you going to know whether what you're testing is working? What's your outcome of interest? Is it just controlled diabetes? Is it still, or is it weight loss? Or is it HbA1c levels? So understanding what your measures are and picking those out, defining them, and then what's the appropriate follow-up time to measure the outcome? Are you looking for a very quick intervention and you expect to see very quick results within a month, 3 months? Or is it going to be 6 months to a year? Also keep in mind the duration of the funding you're applying for. If you're applying for a 1-year grant versus a 5-year grant, know, what kind of experiments you propose need to fit into that timeline. You should not be proposing 3-year follow-up in a 2-year grant. You're not going to have data by the time that funding ends. All right. So once you've defined your research question, you've refined it, you've identified the exact variables, measures, populations, comparisons, outcomes, and time frame, then you have a lot of information under your belt. You should know the literature pretty well. You should know what's going on, what we know about what's going on. So then you form a hypothesis. You need a hypothesis to write your specific aims. This is all driving toward your specific aims page still. So form a hypothesis. And a hypothesis, just as a reminder, is not a question. It is a statement. It's a proposed explanation for something that is not yet proven, but can be tested, right? So we're no longer using question marks. We're saying, maybe if you have enough TGF beta signaling, then you have good vessel wall integrity and nobody's going to die of aortic dissection. Nobody's aorta is going to burst. However, perhaps if you upregulate and have too much TGF beta signaling and somehow something upstream is out of whack and it aggravates and changes that signaling, then maybe that causes a weakness. Too much of a good thing might be a bad thing. And so the hypothesis might be aberrant TGF beta signaling or excessive TGF-beta signaling could possibly have negative effects on aortic wall integrity. That's testable, although I wouldn't do it in live people. Okay. So, like I said, we're driving toward your specific aims and your overall page structure. So, in the blue on the right-hand side, I've got this is your specific aims page, what it should kind of look like. You get one page for it within the specified margins and fonts and all that stuff. All of that's available on the NIH website. The URL is down there. So the top half of the page, generally it's a half a page, could be a little bit more than half a page. I wouldn't recommend this being overwhelmingly most of the page. You need to identify the problem. You need to identify the knowledge gap. You need to explain the current understanding. You need to include any hint, not all exhaustive, but a hint of preliminary results of yours or prior work of other people's. And you need to state your hypothesis and the goal. Identifying the knowledge gap, explaining the current understanding, all of that relies on other people's work, prior work, published literature. And then in the second half of the page, you want to describe your 3 specific aims. And then you want to conclude by explaining the impact to, again, we're writing for NIH, so it needs to go back to human health. Even if you're in the most basic system and basic science, you have to explain the impact as it relates to human health, because that is the mission of the National Institutes of Health. Okay, so things to keep in mind on the left-hand side of the page, all aims should address your overall arching goal and hypothesis. You should be asking a question, forming a hypothesis, and your 3 aims are designed to independently address, answer, either support or refute the hypothesis and goal that you've listed above. Your aims should never depend on each other. So you should not say, I'm going to do specific aim 1, and depending on those results, if those results turn out the way I want them to turn out, then we will use those results in specific aim 2. Because chances are you're not going to get the results that you want in specific aim 1, which negates the rest of your specific aims, and then your whole thing is null, and that will likely get rejected, quite honestly. So if you're going on a fishing expedition, do it in specific aim 3 and keep 1 and 2 for answering very specific questions. So they should never depend on each other. And the scope of the aims, again, should match the duration of the funding and the number of people doing the work. So you can't cure cancer in a typical 1-page specific aim, typical R01 grant. You just can't. You're not going to. And keep that in mind, because reviewers don't like that when you're overambitious. Okay. So going a little bit deeper, what to include in your specific aims. Remember, it's just 1 page. You need to acknowledge the variables, measures, and unknowns that you're going to look at. You need to acknowledge your controls. What's your comparison group? You need to talk about sample size and sample size calculations and your statistical analysis. No one-hit wonders with results. This needs to be, you need to define your results, define success, and then define how you're going to know it's successful. And you need to acknowledge any potential bias and confounders. And then alternative approaches in case the entire thing blows up. It's not going to just blow up. You're going to salvage it. You're going to learn something new. You're still going to push the field forward, but it's about how you're going to use what you find to do that. Okay. And then for those of you who don't play Monopoly, this is from the game Monopoly. But you need to pressure test your specific aims page. Your specific aims are going to take you a lot longer than you ever want them to take. Because what's going to happen is you're going to write the top half of your page to justify your research, and then you'll write out your specific aims, and then you'll have 5 people review that, and they will ask as they review it, are these 3 aims that are proposed going to answer the question address the hypothesis that's described in the top half of the page? And 4 out of 5 people will come back and say, I don't think you're going to answer your question. And then what do? You're not going to keep writing. you're going to go back and you're going to review the literature, and then you're still going to you're going to refine your question again, and then you might have to refine your hypothesis, and you might have to redefine which gap in the field you're going to look at. But you need to then make sure the experiments that you tweak are going to answer the question you want to answer. And this becomes I see this all the time. You will do this. Trust me. It's not everybody will go through this. You will define a question and then propose experiments, which then somebody will point out to you will not answer the question. So then you will start answering the question, or you start adjusting the question, but then you're actually going to also adjust the experiments. And this is why you should never continue writing until this is pressure tested and everybody feels good about your specific aims. Because as everything changes, you're just going to be spinning your wheels if you write anything about, any other part of the grant. So start this now, start it early, and get as many people as you can to look at various drafts of this, and get them really, really critical people, the most critical people and ask them to review your specific aims. And have conversations with them, ask them to review it critically, take that criticism. It's not an ego thing. It's not about you. It is about how you understand the question, how you're proposing your hypothesis and experiments, and whether or not there's logic that makes sense, and that your proposed experiments will generate results that relate back to and sufficiently answer the question. Okay, so there's an art to just doing this one page, but trust me, if you get this one page down, when you get this one page down, and again, it will take you much longer than you think, but when you get it down, you are good to go to write the rest of it. The rest of it will flow. Okay, so get used to this cycle. Don't be daunted by it. Know that you're going to go through this, and know that you're probably going to go through this a number of times. All right, so once you get past the specific aims, once that is nailed down, people feel good about it, not just you, but other people, your mentors, your trusted advisors, then you start working on the research strategy. Now, NIH page limits, this is the body of the grant. So you're to put together a research summary abstract that's no more than 30 lines. I've seen so many of these truncated, people use weird fonts and weird margins, and when NIH puts them up on Reporter, the last few sentences are always cut off. So stick to 30 lines, stick to their suggested fonts, stick to the margins that are required, and maybe aim for 29 lines. Your project narrative is only 3 lines, and sometimes that is less than 3 sentences. Then if you're doing a resubmission or a revision, you're allowed 1 page to address the reviewer's comments. And then you have a 1-page specific aims, which we just talked about. So the rest of it, the body of the grant, the actual proposal, is the research strategy. And there's 3 sections to that. The significance, the innovation, and the approach. The page numbers in the parentheses are what I and my colleagues recommend to early stage investigators to try to aim for as you're writing your first few grants. The reason is that you'll see in a bit, significance and innovation actually count for a huge part of your score, and should not just be 1 or 2 sentences. Okay, so we'll talk about significance and innovation. So people get these 3 confused all the time: significance, innovation, and impact. Significance is about the question. Is answering the question going to make waves in your field, in science, in our understanding of human biology, in medicine? If yes, then your question is significant. Innovation is about the solution. How are you finding the answer? Are you just repeating the same old assays that were developed in 1963? Are you applying tools that have been developed for aerospace engineering in biology? So how are you answering the question, and how are you finding that solution is innovation. The more innovation, the better. Impact is whether or not it's going to make a difference, again, for NIH, for human health. If you're writing for a foundation, and you should also stress impact as it relates to the mission of the foundation. So this is just a chart. You'll get these slides to keep all three separate, and make sure you're writing, when you're writing significance, you're writing about the question. Why is it important? How is it going to change? How does this question might change our understanding? This needs to convince your reviewer that this question is actually worth asking, that they want to give it money to find the answer. Okay. Innovation. How are you coming up with a solution? Do you have a clever way of coming up with new answers? This section really needs to convince the reviewer that your work will shift how we answer questions in this field, right? It'll change how we address questions. It's like applying the human genome sequence to the field of genetics. I was just talking to somebody the other day, the heyday of human genetics and doing, tracing back families and all that, and looking at markers and all that. That's kind of gone. We've applied genomics and informatics to all of that, and it's great. But that was paradigm shifting to how we look at human genetics and congenital defects and diseases. And then again, the impact, always bring it back to human health. How is this going to impact our understanding of human health? How's it going to improve human health? How is it going to change the patient experience? And that impact part goes at the end of your specific aims. Your significance and innovation go in the body of the research strategy. Okay, so I always find that it's most useful for me if I know what people are looking for. It's kind of flipped because I know you're very into your own work. You know it really well. You could sit down and probably write out everything you want to do, but it's not about that entirely. It's more about what the reviewers are scoring and how they're scoring. And like I said before, these are new review criteria effective just earlier this year. There's 3 scores you get in an R1 grant application. They kind of vary. They will vary from a K award to an R award, but in general, by what kinds of scoring is done. But in general, they're looking at the importance of the research, which is addressed in your significance and innovation sections. And remember, that's like 4 to 6 pages of your whole 12-page research approach. So that's significant. That's a chunk. And then they're looking at other 2 factors, which we will also talk about. So significance, what you need to include. Significance is the project's ability to address an important problem or a critical barrier to progress in the field. You need to present the scientific premise and the importance of your research in the context of current scientific challenges, opportunities for advancing knowledge. And that means remember, you're the cog, you're the tiny little golden cog, and then there's a whole world of cogs around you. So how is your work going to bridge knowledge gaps in your field? What is that problem you're going to solve? What is the conceptual or technical advance you will make in solving that problem? You need to address the importance of your research in the context of the funder's priorities. It sounds like I'm repeating myself, but I see so many drafts where people only write about what they're going to do and their work and ignore the world at large. And they come back because there's a weak significance. So address your work in the context of others. And also what you need to do is acknowledge the rigor of prior work. So what were the previous papers missing? What did people not answer in the past? It could have been a technological barrier that we can now overcome, or it could just be a scientific thing that now in retrospect, we understand the problem in more depth, and it's time to go back and fill out some of the answers, right, to make it clearer. So again, looking at what reviewers want. The reviewers and these are all instructions to reviewers posted on the NIH website. Evaluate the importance of the proposed research, that's what you're writing, in the context of current scientific challenges. Assess whether the application addresses an important gap in knowledge. And I've highlighted at the bottom, evaluate the rationale for undertaking the study and the rigor of the scientific background for the work i.e., did the author address and analyze the prior literature or the preliminary data? So you need to bring this into your proposal as you write it. And that's why I said, as you're looking at literature, it might just behoove you to write a literature review as you're doing that preliminary literature investigation, because that's how well you will need to understand it. You might as well get a paper out of it. So common reviewer criticisms of the significance section: lack of or weak scientific premise. You haven't done a good job explaining your role in the context, your research in the context of what we understand in the field at large. The study will result in incremental changes rather than paradigm shifting changes. Oh, they've only gone back and they're going to answer one more thing about something we know a lot about, and you haven't explained why that one more thing is so important that we can't move beyond that. We can't advance in the field without understanding that aspect that you want to study. Or the study will confirm what we already know. And references are dated and do not reflect the current field. This is why you need to go through the literature often and a lot. Okay, so innovation. Now, remember, significance is about the question, innovation is about the answer. So how does this shift research or clinical paradigms? What are the new concepts, approaches, methods, methodology, answers, knowledge? What is new? What are the advantages over existing approaches? What is new? Any refinements, improvements, or applications of existing techniques? And again, the only way you can say this is new is if you also point out this is old, i.e., what was in the literature in the past, right? So you need to say other groups have shown previously, previous experiments have shown X, Y, and Z. Ours will push this forward. Ours is new, and because we're using these approaches that differ and are better from what was previously done. So reviewer instructions for innovation. The extent, now this is tricky, the extent to which innovation influences the importance of undertaking the proposed research. People struggle with innovation a lot. So you don't have to go inventing new technologies just to answer a common question. The newness of the results potential results also counts as innovation, right? But you have to, as an author, draw out the new concepts, the new methods, the new answers, the new ideas, and the new conclusions that will come from the expected results. And that's the challenge of the innovation. So the most common reviewer criticisms of innovation, that the PI is just taking a standard approach that's already been published to answer this question. And by doing that, they're only coming up we're going to see either what we already know being reaffirmed, or it's just it results in an incremental change. Okay, so as you're writing that, as you're writing your draft, remember that they're going to be looking for the new and paradigm shifting results. Okay, so talk about the approach. We just talked about significance and innovation, and we recommend to our trainees, our new faculty, anywhere from 4 to 6 pages for that significance innovation chunk. And then, so the rest of the 12 pages, really 6 to 8 pages, is your actual approach. The approach is really what you're going to do. I'd say the biggest mistake I see is somebody just sitting down and writing, I'm going to do this experiment, that experiment, then I'm going to do this other experiment, and I'm going to do another experiment. And by the end, we'll have the answer to the question I referred to above. That is not a satisfactory approach. The reason is NIH is looking for rigor. All scientific reviewers are looking for rigor. So rigor and reproducibility are critical to how you think about the work. And that is how strong are the conclusions that we can make from the data that you get, right? How well are the experiments designed that you are certain that what you get is the actual answer and you can conclude something about that. So this is only part of a brochure or handout on the NIH website. There's actually 4 areas of focus. As you see here, I've included the links to these pages. I just truncated it for just to keep it, easy on the eyes. So in looking at rigor, you need to assess, and I've already talked about this, the rigor of the prior research. How good was the prior research? It got published, it was funded, it was great for when it came out, right? So nobody's refuting that. But vis-a-vis what we know now, vis-a-vis the tools we have now, vis-a-vis all the other papers that have been published since then, what gaps can you pick out and improve upon and address to better answer, better understand the question of interest? And then how do you design your experiments moving forward so that they give you equally strong or stronger answers and stronger conclusions? So addressing rigor in research strategy. So you will have already, this top part, describe strengths and weaknesses in the prior research to support rationale for current proposal. You will have addressed some of that in your specific aims already. In your approach, you have to discuss your approach as it relates to identified weaknesses of rigor of the prior research. You also have to talk about your experimental design in the context of what has been done in the past and explain why your methods and approaches will give you robust and reproducible results. You'll have to explain why you choose the variables that you're choosing and how those are factored and why those are the best variables to look at and how they will best answer the question you're asking. So factor 2 of what your second score that you're getting on your grant is rigor and feasibility. Is it doable and is it believable? Right? And this all sits in your approach. The first third of your score was about whether it's important and how new is it. This second part is just, is it believable? Is it doable? Okay, so that's the rigor and reproducibility part. This is a lot, but this is what you need to include in those last 6 to 8 pages of your research approach. You have to have the overall strategy, methodology of your experiments. You need to address why these experiments are a significant improvement to any approaches that have been taken in the past. You have to explain clearly enough the experimental design, not down to the buffer components necessarily, but enough so that the reader, the reviewer, believes that this is a solid experiment and you're not going to get garbage results in the end. You have to acknowledge potential problems and confounders that might arise in these experiments. What might confound the data that you do collect, the results that you do collect. You need to, if you're just launching a new approach to studying a new problem, there's going to be questions about feasibility. So what can you draw upon from your own preliminary work? This is where preliminary data really helps. Or what's been done with similar approaches to other problems that other people have done to minimize the chances of something high risk going totally south. You have to explain your variables and possible hazards and your data management approaches, all in those 6 to 8 pages. So definitely include preliminary data in your approach section. Whether it's unpublished, in press, or just an observation in the lab thus far, it helps to make the case for feasibility in what's being proposed now. It's a must. There are very few grant mechanisms for which there's no preliminary data required. And even the ones that say preliminary data is not required, the norm is that people are submitting some preliminary data. So as far as the approach section, reviewers are asked to evaluate the scientific quality, that's not a surprise, to evaluate the likelihood that compelling and reproducible findings will result, okay? So, and to assess whether the proposed studies can be done well and done within the time frame proposed. Again, getting back to the length of the funding cycle. Is it possible? Or is it not possible? Or do they have questions about it? If you're writing in a way where people are like, I don't know, they're going to run into some problems. It might take longer just to figure out the technology or the approach or the proof of principle. That's going to get you an impossible check on this, and you're not going to score very high. So, reviewers are asked to evaluate the potential to produce unbiased, reproducible, robust data, whether the appropriate controls are in place, whether the sample size calculation has been done, whether you're looking at enough N, basically, whether the sample size is well justified, and then how you're going to also, this also goes in the approach section, once you have the data, what are you going to do with it? How are you going to analyze it? Are you going to use statistical analysis? How are you going to judge that it's the right data? How are you going to judge that it's accurate data? What are you going to do to believe your own data? And what are you going to do with it moving forward? So reviewers are also asked to evaluate on feasibility. Is this approach sound? Right? Are you balancing all your blocks on all your Jenga blocks on a single vertical one? Or does it have a more solid foundation? Are you sure to have it work? We just don't know what the results are. What are your plans if this whole Jenga structure falls down? How are you going to salvage what you do find into new findings and new understanding, and maybe course correct for the research approach or the question that you're asking? If feasibility is less certain, is it higher risk, high reward? How are you going to explain that, right, in a way that gives the reviewer some comfort level that, okay, even if it doesn't work exactly as predicted, projected, hoped, they're still going to get something out of it, and it still will be useful to the field. You need to have explained all of that and acknowledged it in a way that makes that reviewer comfortable enough to score you well. So this research strategy is the heart of your application. It will be dense. You will not have enough room to say everything you want. You will want to talk more about the details of your project, but you will need to keep that measured and spend a lot of time referring back to assessing the rigor and how comparing what you're doing to what's been done before to assert that your approach will result in rigorous and robust conclusions. So do add the preliminary data, because that bolsters the evidence that this will work, that what you're proposing will work. You can add it as a preliminary data subsection before you write the approach, but it cuts into the page limits anyway. It's preliminary data fits in that, and it takes up space. So you can tell them all the preliminary data at once and then write the approach, or you can work in the preliminary data as it relates to each aim as you're describing it in the approach. Highly recommend creating an outline and subheads. They take up room, but it helps your reader keep track of where they are in the proposal, and it helps the reader stay on track with your logic. We'll talk about this using tables, charts, and figures to illustrate, clarify. They also take up room, and you have to make it worth it. Otherwise, they're using space that you can't afford to use. And then a note: URLs and hyperlinks are not allowed ever in any part of the grant. There's one exception I will talk about in a bit, but unless the funding opportunity announcement asks for hyperlinks, don't use them. Don't use them. I don't know if they get kicked back or they just get ignored. Best case scenario, they get ignored. Worst case scenario, you add hyperlinks and your grant gets kicked back without being read. So don't do it. There's one exception. Talk about it in a bit, but it doesn't go in this section. Okay, BioSketch. this is about you. So there's two types of BioSketch templates. There's a non-fellowship and a fellowship type. I'm going to talk about the non-fellowship because this is really about you getting out the door, launching your own career. This is the one you're going to need to use for the rest of your life. It will change format. It was about to change format, the requirements this May, in fact, but I think they've put that on hold. So we're still using the piece of paper. Make sure up here on this form, the OMB number, make sure that you're using the most recent BioSketch template. I see this often in that older investigators will pass along their BioSketch. Younger investigators will be like, oh, this is a good template. Let me just cut and paste and, replace all their stuff with my stuff. And then it's an old form and it gets kicked back because your administrative details are not up to date. So, because you use the wrong form, which really sucks. So don't do that. So make sure you're downloading the accurate form, the most up-to-date form from the NIH site. So this is what it looks like. It's just a blank piece of paper and this on top. You fill in your name. You have to have your ARA Commons ID, your position, your training, and then your institute and location. There's 3 sections below that I'll talk about in a bit. Those are free form. What reviewers are looking at. So this is a third part of your total grant score. First part, again, was significance and innovation. Is it important? Is it new? The second part is it robust and is it doable? And the third part is who's doing it, right? And this is the BioSketch. So I'm not going to talk about environment. Environment is going to come from your home institution, who's on faculty, what experts you have, what core facilities, what services, and what tools are available to you. How much support do you have from your department, your institution? All of that feeds into environment. The BioSketch is investigators. So do you have the right expertise to do this work? Do you have the right expertise around you to help you do this work? Does anybody know what they're doing at all? And hopefully the answer is yes. So BioSketch limit is 5 pages per person. You will not fill 5 pages as an early stage investigator, I don't think. By the time you're senior and established, you will have a challenge getting everything in within 5 pages. So it will grow as you grow, and then at a certain point, it will seem limiting. Right now, it shouldn't be. So the 3 parts of the BioSketch that are free form, your personal statement. This is a narrative told in the first person. I have training in molecular biology and genetics. I have also had a longstanding interest in teaching, and I have had experience in communications. And this is why you are well suited. You should amend your personal statement for every grant. I see too many times people doing, especially the big training grants and the big collaborative grants, people just submit an old BioSketch. Some of the more established investigators have an educational BioSketch where they talk a lot about their training expertise generically, and then they have a research BioSketch, and they just send that in randomly, and it ends up not being entirely aligned with the focus of the grant, and that can ding the grant, quite honestly, if you don't spend the time. So make sure as you're starting out, you will write or edit or revise a new BioSketch for every grant you apply for. This is also where you can highlight experiences and skills that aren't obvious in the rest of the BioSketch. So the personal statement part is very important and really needs to align well with the whole purpose of the grant. It needs to show that you're the right expert, you have the right interests, and then everybody else who's part of the grant also needs to submit their BioSketch, and they also need to have relevant and focused expertise toward the goal of this grant. Part B is positions, appointments, and honors. This is kind of a timeline of your career, your positions, and any honors and notable advisory roles that relate to, especially for research BioSketch, you want to have research awards and commendations. For a teaching, training grant BioSketch, if you're going to be part of that training grant, you want to have teaching awards and commendations. You shouldn't mix them up. They shouldn't overlap. You should not be using the same BioSketch for every grant you apply to. It's just not generic enough. I mean, it doesn't work that way. And then the part C is contributions to science. You're allowed to describe 5 key contributions. with a brief like paragraph description of each, and you're allowed to point out and note the citation. You're allowed to list the citations of no more than 4 relevant publications for each of these contributions. Now, as you're starting out, you may not have 5 related contributions. It's okay to only use 3 or 4. It is not okay to put in 5 contributions for a grant, but 2 out of the 5 contributions scientifically, topic wise, have nothing to do with the grant. That is distracting, and reviewers will not be happy with that. This is the only part of the NIH grant where a URL, a hyperlink, is allowed. You are allowed to put in your PubMed link at the end of your contributions to science to link to your, whatever search term you use, whether it's first name, last name, some combination of your initials that gives the person who clicks on it your PubMed, the results of your PubMed search. Okay, common weaknesses in biosketches. Weak personal statement. Generally, it's too generic. Somebody's just written a first-person story about their whole career path and not pulled anything out because they decided to do a generic one. It's not related. There's no mention of the grant being proposed. There's no mention of anything specific, and it just fails to, it like floats on its own. It doesn't really say, hi, I'm the right expert to do this work. Lack of relevant expertise for the proposed research. Again, the contributions to science are not related to the proposed research, or sometimes people write, really technical contributions to science and nobody can understand what the hell they're talking about, pardon my French, and then the reviewer can't judge whether or not they actually have the expertise to do the work that's being proposed. Also not good. Too vague, this especially comes up in multi-PI or co-PI situations where you're 5% on somebody else's grant and you just put down co-PI. No, what were you responsible for doing? What did you do? What did you generate? What were your roles? Those need to be stated, and they have to be also related to the grant currently being written and compiled. And then people use old biosketches and they never update their publications. Things have been in press for the last 5 years, and that just don't do that. These are easy editing fixes if you just spend a half an hour on your biosketch each time you apply for a grant. Just make it good. Okay, last thing, figures. Everybody loves figures. Why do we use figures? We use figures because the written instructions for putting up shelves are horribly boring, and you've got tools and safety goggles on. You cannot read this fine print. So IKEA discovered that you don't even have to use words. If you just use the right pictures and illustrate instructions, then everybody can put the thing together and in any language they speak. And that is the hallmark of a well designed figure. Okay, but we're not IKEA. So use figures. Remember, figures take up space. So you should use a figure when it's needed to enhance or clarify what's already written. You've got a complex molecular pathway. There's a lot of things going on. Or, you've got a conceptual model of something that's happening. There's arrows and boxes and inputs and outputs, and a nice flow diagram, a simple diagram there can really enhance what is written. Okay, so let's just do that. Present a clear message and a clear process, right? A nice flow chart. Data visualization. I'll come back to this. I would say use that very sparingly. Conceptual models, workflows, timelines work very well. Figures. Don't just use a figure because you think the page has too many words on it and needs to be broken up for the eyeballs. Don't use figures just to make your application pretty or whatever that means. I don't even know what that means, but I've heard more than one person say that. So I'm saying it to you. That's not a good reason. Don't explain concepts. Don't put a figure in when you can't even explain it in words. And certainly don't put a figure in and not explain it in words. Don't substitute words with a figure. That's bad. So how and where, when and where to use them, read the instructions very carefully. There are many places in a grant proposal where figures are absolutely not allowed. They're not allowed in the specific aims at all. Don't do it. They're not allowed in the project summary abstract. That's the 30 lines that everybody always writes too long. The last few lines get truncated. That gets stuck on Reporter, NIH Reporter. A figure will just mess up the whole thing and they won't be able to post your thing. It's an administrative mess. Don't put it in the project narrative. That's 3 lines. It doesn't need a figure. Don't put it in your biosketch. You don't need a picture of yourself there. That's all words. Don't put it in a letter of support. We didn't even go over that. That's what other people write. Don't use figures there. Just stay to words. So there's really, it's your research approach, research plan section where figures go. So how to use them. Identify key concepts or data points that, if you illustrate them, will make it so much clearer to the reader. Don't make a decision like, oh, I need a figure on every page. No, that's a bad approach. Only for the tough concepts or the concepts where like just having a simple figure will be like, oh, okay, now I get it. Focus on ensuring your reader understands the overall big picture, your conceptual model, what you think is happening. Make figures specifically for your grant. Do not take a figure from a published paper and plop it in your grant. Use color strategically. You don't have to use color. Sometimes it works better in black and white, and always keep it simple. Okay. So figures must provide value added. What I have on the upper right is a figure from a real grant. This actually was funded, this grant, but this is kind of at real size. There are 9 panels to this figure. It is the histology completely useless. Bar graphs are great in a grant proposal. Histology, I wouldn't do. And then lower left, okay, find the Western blot, fine. Immunohistochemistry, I also wouldn't do. I can't tell what's going on in any of these. By the time they're reproduced, I actually blew this up. It was much smaller in the grant proposal. Nobody can see what's going on there. And a reader who spends too much time trying to figure this out will be mad. So distracting, confusing, illegible figures make readers mad. They must be readable when printed on normal size paper. So if you've got it shrunk down to save space, but you really want to include a figure, think about your cost benefit analysis. For this top figure with the 9 panels, I probably would have just picked 1 bar graph as an illustrative example of the data that I've gotten and talked about it and other things not shown and be done with it. Because 1 bar graph at the size that's legible will not make me mad. The histology right now is making me mad. Keep your fonts consistent, keep the figure legends legible. There's all kinds of regulations and rules on the NIH website about this. Keep in mind that color blindness happens. And you might have readers with visual impairments and don't want to make them mad or leave them out. And also color can be distracting and it may appear different on people's devices. So use it strategically. And again, don't make your readers mad. People get very emotional about colors and figures and not being able to see stuff. So don't add that variable to the review of your grant unless you're certain that it's working for you. So just an example of types of color blindness, particularly just red and green color blindness. Just notice, on the left, we've got normal color vision, red and green. On the right is what different types of colorblind people see. And then separately, this figure 2 on the right, the bar graph that's only shaded in green and various forms of red, is also taken from a real grant. And just imagine what that would look like for somebody who has color blindness, right? Like don't do it. Okay. Lots of tools out there. BioRender, I'm sure you're all aware of. Other tools, I'm not going to belabor this. You're probably already using some of these. Lots of things out there that you can use. So just to sum up for today, develop a writing habit. If you don't already have one, start writing. Get your specific aims nailed down before writing anything else. You will be spinning your wheels otherwise. it's not worth it. Spend the time, invest the energy on your specific aims. Make sure they're solid. Make sure that you've explained the significance and innovation, particularly in relation to previously published work, of what you're proposing now. Your approach also must be rigorous vis-a-vis what was previously published and feasible within the scope and size of the funding being applied for. BioSketch. Make sure it's accurate, up to date, and relates to the rest of the grant application. And use your figures judiciously. Back to this. Beg your friends and colleagues to critically review drafts and take their suggestions and comments to heart when you write revisions. Questions?
Audrey, thank you so much. There are a lot of questions, so we'll go through as many as we can. One of the first ones was early on, as you were talking about developing a writing habit. Somebody was asking if you mentioned a specific app to help with timing. I suspect they're asking about the website for Wag Your Work. And so I'll let you chime in if you did have something specific, but I was just going to say we don't have specific writing accountability groups that are put together by OITE. Some institutes do that, and I absolutely encourage you all to do that on your own as well. There is an FAAS.
Oh, you went mute, Laura.
Sorry, there is an FAAS writing course that is happening this summer. It happens regularly, but it looks like the next one is in July. So I've just put that in the chat. That is an option for walking you through the process of writing a paper if you're at that stage. And there are also writing resources through the NIH library as well that I'll put in the chat. Yeah, but Audrey, was there anything else as far as?
So no, there's not a specific app that I use or that I recommend. Wag Your Work is a book, but they also have a pretty extensive website that, kind of explains what a WAG is and how you can set one up. Anyone can set one up, and it's not really, it's an accountability group. So I think you have the freedom to do what you want, but setting the timer, it was my analogy of why Peloton works so well with their app, in that they do offer 5 and 10-minute exercise classes. So it negates any excuse you have about not having enough time to write, because you don't need that much time, right? So as you get started with exercise and writing, start with 5 minutes, but make yourself stop, right? You don't want to run your first exercise out that you don't want to go for an hour. You're going to ruin your muscles. And the same thing, you don't drive yourself to hate writing by making yourself write for an hour the first time you sit down. Write for 5 minutes. Step away from it before you hate it. Then come back to it the next day.
Awesome, thank you. A lot of questions about slides and recordings, so I'll just say that we will send out the slides and the recording for this session and for session 1 to all registrants, just so you all have them. So everybody will get that. We have a couple of questions about, you talked about how to form a hypothesis and how to talk about your hypothesis through your aims. A few questions saying, what if I'm not doing hypothesis-driven research? What if I'm building a tool? What if I'm, going for something that isn't directly testing a hypothesis? How do I approach that?
Yeah, so I think look at the funding opportunity. Make sure it's not looking for hypothesis-driven research. I would start there and make sure you're really understanding the funder and what they're looking for, and you've identified an appropriate funder for the non-hypothesis driven work that you're doing. Because I think it's going to hurt you if you apply generally and widely to funders and opportunities that are looking for hypothesis driven research. So that's step 1. Step 2 is then you really have to, if you're building a tool, then you do have to emphasize the rigor of it, the newness of it, the utility of it, and the impact. So and and rigor and reproducibility, all of that still go into it. You don't need the hypothesis, but you need, you need the use, your evidence for whether or not it's needed. And that still goes back to justifying the significance and the innovation. You just, and you still need an overarching goal. It doesn't have to be hypothesis driven. But again, that needs to match what the funder seeks and the funder's mission.
Awesome. There's this question about, so this is from a current postdoc, but in general, I'm a trainee right now. I'm working on my PI's work. And when I'm writing an early career grant, I'm really proposing different work that I want to do when I start my own lab. How do I approach getting preliminary data for work that I'm not doing right now?
So it doesn't necessarily have to be your data, preliminary data. But I would say, okay, Laura, you froze, so I don't know. Okay, so I would say what's important for doing these early career grants, and especially if you're doing, a K mechanism, it's more about you and the mentorship group you put together and their research and how that supports the research you want to do than it is about the preliminary data. So it could be their preliminary data that you want to take a little bit further, right? But it's also about who you have in that mentorship group, who's supporting you as you basically train more. So don't feel like you have to sneak reagents home and generate your own data on the side for preliminary data. It's about having some evidence that there is traction in the direction you want to go, regardless of who generated it, and that you have the support to pursue that further from mentors and other folks who might help with the resources to do that research. Does that help?
Yeah, I think so. Yeah, thank you. Um, a couple of questions about sort of what's the difference between significance and impact? Where's that line? They seem similar. And then how do we figure out what's going to be interesting to the reviewers? How significant is significant enough?
Ah, age-old question. So let me tackle the easy one. Significance is it important in the field writ large? Impact is how does it come back to the human health, human patient? If you're coming back, if you're writing for a foundation, it's really, the mission of the foundation. How does it impact whatever? Gates Foundation, for example, would be global health, right? American Heart Association, you would write the impact, better cardiovascular health, longer lives, healthy longevity. Significance is it a big meaty question, or is it just like, here's something it would be interesting if we answered it. Life will probably go on if we don't answer it. Or, if there's not enough impact, then it's not going to be significant for sure. Does that help?
Yes, thank you. You addressed this last time, so I'll say this is in the recording from last time that you all will see, but maybe you could just briefly speak to your approach for using AI for grant writing.
Yes. So I love it personally, but I'm not using it for much original writing. The problem with AI is that everything you put into AI is going into the black hole of AI training, and the private companies that are developing these AI models are taking your input and using it to train their models. So that means if you give it unpublished results because you want to turn this unpublished result thing into a well-written paper, your unpublished results will show up in somebody else's search results. So I know that at University of Maryland, we have a Microsoft contract, but we've also negotiated as an institution with Microsoft to have our own firewall. I can't remember what it's called, AI, whatever it's called, the Microsoft AI. So that when we can use it to write our own papers, and the stuff that we put in it is not stolen by Microsoft. That's a strong verb. So I would say be very careful about how you use AI. AI is great for non-native English speakers. If you want to ask it, you know, this is a sentence I wrote. Please make it more smooth. Like it will spit out a better sounding sentence. You should probably pressure test that against a native English speaker anyway. But if you want to do improvement in your diction and your English, that's probably very useful. If you want to take it and ask it ideas from out in the ether, that's probably fine, because you're asking it to look for information that's already publicly available or out there. If you're using it to write your own original papers, I would be very suspect and very careful. So it has its uses, by all means, but understand its limitations.
Yeah, thank you. So we are right at 12:30. I know I didn't get to all of the questions. I am going to compile a document of questions from this workshop and the last one to share some answers. And in cases where I don't have an answer, I'll check in with Audrey too, so that you all get answers to those questions. I want to thank you all so much for coming to the event. And Audrey, thank you very, very much for presenting.
Thank you. I hope it was helpful, and good luck to everyone in your careers. And I hope we cross paths sometime.
All right. Thank you, everyone. Have a good day.